So, having calculated the chemical shift index as we saw in the last class, now the question is how do we show it pictorially, because in the previous class we saw we are calculating it for one amino acid at a time, but in naturally we want to see whether the entire protein, what is the overall secondary structures present in the protein. So, this is where we start plotting the CSI graph or the CSI plots. So, how do we do that? Let us see here. So, let us say this is your protein primary sequence. Okay. So, this is how it goes from one end to the other end. So, what we do is, if our the three chemical shift that we saw, the three different criteria for alpha helix, that is for H alpha should be less than minus 0.1, beta C alpha should be greater than 0.7, CO should be greater than 0.5, so on, then we call it as alpha helix. So, if all the three chemical shifts or 2 out of 3 agree, then we say that that particular amino acid is in alpha helix or not. So, if that particular amino acid, for example serine here is considered as alpha helix, we will assign a number -1 to serine, just a simple number. It is a arbitrary -1 is just a number assigned or given to S, this particular S, if it belongs to alpha helix. If this S according to the CSI approach was found to be in a beta sheet conformation, we will make it 1. So, let me correct this, this is a mistake. So, this should be 1 and this should be 0. Okay. So, if this serine is found in a beta sheet conformation based on this approach which we saw in the last class, then that particular serine will be just given a random an arbitrary number 1. Then similarly loop, if it belongs to a loop region based again on this chemical shift values, we will assign it a value 0. So, what we are doing is simply assigning every amino acid here either 1, 0, -1 based on whether it is in alpha helix, beta sheet or not, which was calculated using CSI method. So, let me show you another example. Suppose I shift to the next amino acid, then here again I will assign for alanine whether it is alpha helix, beta sheet or not. Based on that I will assign one number to or these numbers to this amino acid. So, like this for every amino acid I will assign these numbers and then we will plot it like this. So, this is the plot called the CSI plot. Okay. So, let me write it here. This is called CSI plot. So, this is a very standard plot which is shown in many papers whenever you publish the secondary or a structure of a protein by NMR, this is typically shown. So, you can see here now in this graph, this is for some protein like this which is a ubiquitin. You can see that as we go from one amino acid to other. So, let us say we start from this here on the x axis is the residue number. Residue number means starting from 1 going up to the last amino acid. So, if you see here the first 2 amino acid were found to be in a random coil conformation. So, it was 0 according to this here. Then the amino acid next third onwards continuously they were found to be in beta strand, beta sheet. So, they were given 1 1 1 for everybody, like so. This is shown like this in the picture form. Then again there was a loop, a short loop, random coil, then again beta sheet, beta strand, a short and beta strand and so on. So, look at this stretch here, there was a long stretch where all the amino acids were having negative, means secondary structure was helix, alpha helix. So, they all of them were given the number -1. So, there is a long alpha helix which is shown here, this helix here on the right side you can see, and then again you have random coil, beta sheet, random coil and so on. So, this is how we typically plot the CSI for a given protein to show or depict what are the different secondary structure elements in that protein. Okay. So, this is basically the CSI method which we typically do. So, we will now move on to the next. So, now in recent years there have been lot of course improvements. What I have shown here, this approach was a pretty old one. Now in the recent times people have improved this method to even get more accurate prediction or accurate estimation. I would not call it prediction. Prediction is when you do not have NMR chemical shifts. but if you have NMR chemical shift you can directly identify, and that is published. You can probably go to this website to get a more detailed idea of how CSI today is used for protein structures. So, let us move on to the next secondary structure method, that is based on the J coupling between N and H alpha. This is Hn H alpha, between Hn, that is amide proton, and H alpha proton. So, let us see what is that. Before that, let us also look at what are torsion angles. So, in a protein, if you look at the backbone of a protein, there are different atoms and you can define different torsion angles for different bonds. So, remember, torsion angle is not same as bond angle. So, bond angle is defined between 3 bonds and a torsion angle is defined between 4 bonds, means 2 planes. So, I would recommend you to strongly read this up in a test book on any structural biology, any biophysics or NMR test book, and find out how do we actually define a torsion angle. So, many times people confuse a torsion angle with a bond angle, which is completely not correct, completely wrong. So, therefore, now these are different torsion angles shown here. So, the very important torsion angle for secondary structure is these two, phi and psi. So, these two together help us to tell whether this amino acid. So, if you look at this, this is our amino acid I. This amino acid is alpha helix or beta sheet based on these values. But in NMR, we normally measure only this value, we do not measure this value, we measure only this. And how do we measure this value? We measure this value based on this coupling between this proton and this proton. So, if you notice, this is a 3 bond separation between the 2 protons. So, we use this 3 bond separation, we call it 3 bond J coupling between Hn and H alpha. So, we will see now how we measure it and how we use it for finding the secondary structure of that amino acid. So, you can see this is more 3-dimensional picture of how the torsion angle is defined. So, you can see this is a rotation around this bond. So, if these two groups here, this set of atoms and this set of atom, if they rotate around this bond, then that corresponds to a phi rotation of this torsion angle. Similarly, psi is shown here. So, one has to have a very firm grasp or firm understanding of what is this torsion angle, how do you define them. So, as I said, we will use this 3 bond J coupling and we will measure this J coupling in proteins, and then from there we will try to find out the value of this phi angle. Okay. So, this is how, as I said, the phi is a torsion angle between 4 atoms. So, what you do is you can imagine these 3 atoms as one plane, you can see this dotted plane here, and another 3 atoms another plane. So, when you take these 2 planes, it looks like a book, an open book, and the angle between this open book is defined as a torsion angle. This is some torsion angle, do not think of it as our protein torsion angle, any general torsion angle is defined in this manner. Now, typically in proteins and amino acids, this value, this JHN H alpha value will be of the order of 1 to 12 hertz. Okay. So, this is a number which we should keep in mind, because this is the range one should know for what is the value of a coupling for 3 bond. This is called a vicinal coupling. Okay. We have seen in the first part of this course what is called a geminal coupling, which is 2 bond, and vicinal coupling. Now, this vicinal coupling constants, that is 3 bond, are very well known, related, very well known to the phi or the torsion angle for that. So, for example, let us say I measure this J coupling between these 2 atoms. Okay. So, this is the 3 bond J coupling. So, if I measure this 3 bond J coupling between this hydrogen and this hydrogen, that J value will be related to this phi value by this famous equation, which is known as Karplus equation. Okay. So, now if you look at, if you plot this graph J versus phi, it has kind of this nature, and you can see that it is not straightforward, because for a given J, let us say my J value is somewhere here, I can have multiple phi values which will have the same J values. Okay. So, it is therefore important, for example, if I have a J value of 5 hertz, it can actually be based on either it can be here, it can be this value phi, this can be this, or it can be this. So, there is not easy to find out the phi value straight away from j value, but still we get an idea that where the phi value could be, what kind of values we can expect to get. So, that is how we use this method for based on j value we try to estimate what could be the phi value. So, this is kind of an approximation, or vice versa, if I know the phi value I can estimate what is the j value. So, this is another option to use this approach. So, these are the set of values which tell us different secondary structure. So, you can see as I was mentioning parallel and beta anti-parallel sheet, there is a slight difference. So, if you look at this now, let us start from the helix. If the j value is somewhere like 3.9 hertz, this is an average of course, do not think that it will be exactly this value, it will be somewhere between 3 to 4 hertz. In that case our alpha helix, the secondary structure could be alpha helix for this residue. If the value turns out to be somewhere like 4 or 5 or 6 hertz, sorry 4 to 5 hertz, then it will be 310 helix, and somewhere between 5 to 7 or 8 will be random coil. Okay, the random coil is not shown in this table, but if you get a value of 6 hertz or 6.6 somewhere between 5 to 7 hertz, you can think of that particular amino acid is in the random coil conformation. Then very high value, high means towards the higher side 9 or 10 hertz tell us that they are in the beta sheet. Now, there is a small distinction between alpha helix and beta sheet, sorry anti-parallel and parallel, similar to distinction between alpha and 310 helix. So, you can see this typical average values again, if the coupling constant is very high close to 10 hertz, it will be somewhere can be thought of as parallel beta sheet. And these are the typical phi values which are known in literature based on the 3-dimensional structure of proteins. So, this is based on the high resolution X-ray structure. People know that for an alpha helix what would be the average phi value and so on, that is now shown here, and it is correlated with the J coupling values. This will also follow the Karplus equation which I showed in the previous slide. So, the question now is how do we actually measure this J coupling by NMR. So, J coupling by NMR is actually done using an experiment known as 3D HNHA. So, this is typically done on the N15 labeled proteins. So, what are we doing in this particular spectra? In this particular spectrum or in this particular experiment, we correlate the nitrogen amide and this two atoms, that is N15HN, that is HSQC, so HN H alpha, we correlate that to the H alpha of the same amino acid. So, this is the point here, we are correlating, chemical shift, or we are connecting the chemical shift of the HN, the N15 and the H alpha of the same amino acid. This is very important point because we do not want to go like in HNCOCA where it correlated with the previous amino acid, this is within the same amino acid. Now, the spectrum will come something like this. That means, in a 3-dimensional manner we have seen this kind of a picture for other 3-dimensional experiments. So, a 3-dimensional experiments consist of planes, okay, in the third in one of the direction, and other two plane in this dimension. So, this is a collection of planes along one dimension. Okay. So, collection of 2D planes is a 3D. So, this is something we have been seeing repeatedly in the previous part of the course. So, in this experiment HNH alpha, for any given amide proton to nitrogen pair, so this is like HSQC pair, for any amino acid in the third dimension you will see two peaks. One is called the diagonal peak, diagonal meaning, okay, so let me write the correlation. So, it is shown here. So, this is basically, this is called the diagonal peak. Why? Because this and this are same, this is also HN and this is also HN. So, both are same. So, this is kind of a diagonal peak, okay, but this is a cross peak, and each amino acid like this will be separated in 3D plane by based on their N15 value. So, this is a 3D experiment. So, it helps to reduce the overlap. So, you can see here now for every plane, for every amino acid, I will get 2 peaks for a given NH N15 chemical shift, and that is basically the HNHA. So, again if I want to make it more clear, let us say that I have a 2D HSQC spectrum. This is HSQC 2D spectrum. If I will get peaks like this in HSQC. Now, in a HNHA, if you go in this dimension, and I will get 2 peaks here. Similarly, for some other residue I will get 2 peaks in the 3D. So, this is in the 3D HNHA. Okay, 3D HNHA. So, in a 3D HNHA, we for every peak in the HSQC, so this can be labeled as HSQC. So, for every peak in the HSQC in the third dimension, I am getting 2 peaks like shown here for a given HN plane, that is for a this is like one plane, this is another plane, this is third plane, and there has many planes. So, in a given plane for a given residue I will get like this. Now, using this these 2 peaks we will see now how we will calculate the HNH alpha coupling constant. So, this is what is shown here, this is the HNHA coupling constants, I am sorry, the HNHA spectrum. So, what is done is we do like this. So, this is for a one particular plane. So, let us say I take some plane, I take this plane, but of course, in this plane I have shown here only one here, but it is possible that you may have some other amino acids also in the same plane. Okay. Now, you go for each of this amide. So, this is each one of them is an amide residue, amide chemical shift. For each of the amide chemical shift, you will see 2 peaks like shown here. Okay. So, one is the diagonal peak, you can see it looks like a diagonal, it is going like this, and another is a cross peak. Now, cross peak corresponds to the H alpha chemical shift value, and the diagonal peak corresponds to the HN chemical shift value. So, you will see this will somewhere come around 7 to 8 ppm, whereas this H alpha, this whole zone, this zone is the H alpha chemical shift, somewhere from 3.5 to 5 ppm. Okay. So, this is the alpha H alpha chemical shift values. Now, what we do next is we actually integrate, means we calculate the volumes of these 2 peaks. So, that is what is shown here by circle. We integrate, get the volume intensity of this peak, the cross peak and the diagonal peak, and then we take the ratio. So, when you take the ratio of the cross peak to the diagonal peak, then it is related by some formula to the J value, and this is something which I will not be able to derive in this course, but if you look at some advanced NMR test books, you will be able to see that in that test book. So, what they is this comes from some equation from the pulse program as well, from based on what is called as a transfer functions. So, it turns out that if you take the intensity or volume of the cross peak, divide it with the diagonal for the same amino acid. Okay, so, look here we are looking for the same amino acid. So, for the same every amino acid we take the cross divided by the diagonal, and from that value we will get that is corresponds to tan square into this, or tan square of this, sorry. Now, what I can do is I can invert this, means from based on the intensity I can then take a square root of this and take tan inverse. So, let me write down here. So, if I know this value, okay, so that value I can take a square root of that ratio, I will call it as a ratio, okay, and then I take a tan inverse of this. I am just inverting from here to here, and then divided by 2 pi into delta. Delta is some delay in the pulse sequence, which is not mentioned here. There is a delay value in the pulse program, which I did not show in this one. So, that delay value is delta. So, if I take this tan inverse and divide by this tan inverse, sorry, I have to take a square root first, and then take a tan inverse, and now here you see there is a negative sign. So, your doubt will come why this is. So, what happens is this intensity is actually negative, this is negative intensity, this is positive. So, what happens is when you take this ratio, the ratio will be negative, because this is negative intensity, this is positive. So, that this value will be negative. So, when this is negative and there is a negative sign here, that will cancel, and therefore, directly you will be able to take the square root, because square root will be of a positive number only, because the negative sign of here will cancel with the negative sign here. Now, once you do that, you can now take a inverse and calculate the j value based on this formula, that is you take the square root and then tan inverse. So, this is something which is routinely done, and this kind of approaches in NMR in general. So, this is a very general terminology called quantitative J correlation. So, there are two ways to measure J correlation in NMR. One method is to look at the peak splitting. For example, if I have a doublet, then I can use this difference and calculate J value, right. So, this is called a simple spectral based J estimation, but here we are not doing that, we are not taking the distance between this, we are taking the ratio of the intensity. Okay. So, this is like a quantitative approach, this is not this is also quantitative, but here we are using the separation between the doublet to measure the J coupling, but here we are using the intensity of the peaks to measure the J coupling. So, this is a very fundamental difference between the quantitative J correlation and other types of. So, this is how typically in a 1D we measure like this, in a 2D we can measure with a contour. Okay. So, this is how J values can be measured in a 2D, but in NMR remember there are two methods, we either measure it from the spectra or we can measure using ratio of intensity. There are many experiments published in this area and similarly using this approach. So, you can also use J, you can also measure HNHA J value using this approach, but this technique we have not discussed here. The one which I discussed today was a more popular experiment which is based mainly on quantitation, by taking the ratio of the intensities. So, this basically brings us to the kind of the secondary structure, and as we move on we will see in the next class we will basically look at how now we can move from secondary structure to 3D structure. So, there we will now start looking at NOE based methods and find out the tertiary structure of the protein.